Let's talk about cells. All right, so early cell theory established in 1800s, in the early 1800s. The first tenet is that cells are the fundamental units of life. The second tenet is that all living organisms are composed of cells. And the third tenet, which is highly debated at this point in time, is that all cells come from pre-existing cells. So we have lots of different types of cells. Two of the major cell types are going to be prokaryotic cells and eukaryotic cells. Bacterial cells are a type of prokaryotic cell. If you were to break down the term prokaryote, pro meaning before, and karyote meaning the true seed. So prokaryote is before the true seed, that true seed being the nucleus. Bacterial cells, as well as archaea, actually do not have, there are types of prokaryotes or prokaryotic cells, and they do not contain nuclei. However, all of our eukaryotes have the true seed. They're all going to have nuclei. So things like our skin cells, nerve cells, egg cells, which eventually proliferate and become multicellular organisms. Same with your plant cells. All these are multicellular eukaryotes, though there are still some single-celled eukaryotes that exist. And the reason for all of this diversity is because of evolution through natural selection. So we have two different types of cells, and if you remember, your prokaryotes are not going to have nuclei or other membrane enclosed compartments, or what we would call organelles, and that's going to entail or include our bacteria as well as our archaea. Now our eukaryotes are going to have nuclei as well as organelles, and that's going to include your protists, which are going to be typically single-celled eukaryotes. We have fungi, we have plants, and we have animals. Some fungi, like yeast, for example, are a type of unicellular eukaryotic cell. So if we look at the two different cells here, this is contrasting the difference between a prokaryotic cell, in this case it looks like a bacteria, and a eukaryotic cell, like an animal cell. Now this isn't to scale because, as we might remember, prokaryotic cells are actually much, much smaller than eukaryotic cells. In fact, they're roughly the size of a single organelle that you would normally find in a eukaryotic cell, kind of like a mitochondria. So some things that your prokaryotic cell is going to have are cytoplasm. They're also going to have cytoplasmic membrane, so the cell membrane. They're also going to have a nucleoid region, so they don't have nuclei, but they still have genetic material, and that's all kept within their nucleoid region of the cell. And then they also have ribosomes. Typically, all of your prokaryotes are going to have ribosomes as well, and that's, if we remember, that's going to be the structure that actually creates proteins. It translates your transcripts into protein. And then our animal cell, we're going to notice here we have ribosomes in common. We have genetic material in common that's going to be kept within that nucleus. We're also going to have the, where is it? The maybe it's not labeled here, but we have the cell membrane as well as the cytoplasm in the eukaryotic cell. So those are the things that they have in common. So all cells are going to have a cell membrane. So that's just going to be that lipid bilayer structure that separates the inside of the cell from the outside of the cell. All of our cells are going to contain some form of cytoplasm. So that's the juice within the cell containing the pulp. So that's the medium suspending all of the parts inside. And then all of our cells are going to have some kind of genetic material, whether it's in the form of DNA or RNA. And then they're all going to have ribosomes, which again, those are going to be those structures that create proteins, that translate mRNA into protein. And then lastly, everything is going to have some kind of metabolism or the use of some kind of energy currency like ATP. So let's look at the cell membrane, otherwise known as the plasma membrane. You're typically going to find that in every cell. and it has the model of a fluid mosaic. Fluid because all of those little phospholipids are constantly moving and interchanging with each other in a very fluid way. And then it's a mosaic because it's actually composed of many parts. So things like carbohydrates, lipids, other types of lipids besides your phospholipids, like cholesterol, as well as proteins, which could compose, channels or even signaling molecules combined with carbohydrates on the surface of your cells. Some characteristics of your cell membrane are going to be that it is selectively permeable, meaning some things may pass through it and some things may not pass through it. Typically, the things that are barred from passing are going to be larger molecules and specifically large polar molecules, which will typically just bounce off the membrane or hang out around those phospholipid heads, which happen to also be polar. Now, smaller molecules can typically squeeze through, and especially if they're small and nonpolar, they have a pretty easy time making it through that cell membrane. Another characteristic of your cell membrane is oftentimes they'll be involved in signal reception, whether that's receptor sites for ligands in the extracellular space, or just simply molecules that can interact with other cells, for example. Structures like glycoproteins or different types of carbohydrates that could serve as signaling molecules to friendly cells or neighboring cells. Your cell membrane is also going to play a role in maintaining homeostasis. So it's going to be constantly exchanging things into and out of the cell, whether through facilitated diffusion or active transport or just simple diffusion. And then lastly, it serves as a connection. It could be involved in connecting to other cells in a way that it is physically connected to other cells or in a way that it is signaling to other cells. So all of your prokaryotic cells are going to have a plasma membrane, as we've mentioned earlier. Now, all of their DNA is going to be concentrated in what we would call a nucleoid region. So it's just going to be if you're looking at a prokaryotic cell, it's just going to be, if you were to stain the genetic material, it would just be a highly concentrated area of DNA or genetic material. And then they also have ribosomes. Those are going to be the protein production sites. Those are going to be made out of RNA as well as proteins. Remember, they're made out of a structural or functional, you could say functional nucleic acid as well as protein parts. And then we also have cytoplasm. So all of your prokaryotic cells are going to have cytoplasm, and that's just going to be the membrane enclosed region, which contains all of that juice and pulp within the cell. Now, the juice without the pulp, we call that specifically cytosol. So cytosol is going to be that liquid portion. Now, some prokaryotes, but not all prokaryotes, some prokaryotes may have what we would call a capsule, and that's an outermost slimy layer made out of polysaccharides. If you remember from our macromolecule section, those are going to be types of sugars, so larger chains of sugars. And so when I take my hand and I smear it on my desk, all of the bacteria that are becoming stuck to the desk, for example, are actually sticking to the surface because of their capsule. Next, some prokaryotes may have a cell wall, and that's going to be typically made out of peptidoglycan, and that's going to be located just outside of their plasma membrane, kind of like plant cells. Plant cells usually have their cell walls just outside of their membrane. Now, another fun thing that they might have, if you're a bacteria, is an outer membrane. So that's an additional membrane on the outside of their cell wall. So plasma membrane on the outside of the cell wall with lipopolysaccharides. So we have the cell membrane and then the cell wall over that, and then outside of that is the outer membrane in some bacteria or prokaryotic cells. Now, some prokaryotes will also contain chlorophyll, and if you remember, chlorophyll is going to be that photopigment involved in photosynthesis, and therefore some bacteria could actually photosynthesize. That chlorophyll that they're going to be using will be infolded into the plasma membrane. And we're typically going to find chlorophyll in cyanobacteria. Next, we have flagella. So some bacteria might have a flagella, and if we remember correctly, flagella are actually whip-like tails, small whip-like tails that are made out of microtubules. So they're tiny tail-like locomotory structures used for movement. Now, some bacteria will also have pili, which are going to be these thread-like structures that will help bacteria adhere to one another during mating. So when they undergo the horizontal gene transfer where they shoot some plasmids over, they use pili for that, or they can just latch on to other cells for food, for protection. And then lastly, some bacteria, some prokaryotic cells will have cytoskeletons, which is just a protein scaffolding within the cell. So now let's jump over to eukaryotes a little bit. So about 2 billion years ago, that's where prokaryotes were dominant on Earth. And then about 500 million years later, that's where eukaryotes show up. And their main thing is compartmentalization. They have all these organelles which help with compartmentalization. And what is compartmentalization? That's just allowing you to create little compartments where certain cellular processes happen that wouldn't happen elsewhere. So you're going to have certain cell processes happening within these little organelles within the cell. So they're able to compartmentalize in two different ways. The first one is infolding, so that's just going to involve your endomembrane system, which we'll talk about in a future video. So that's where the plasma membrane folds in on itself, and that's also responsible for forming that nuclear envelope, which we'll talk about in a future video. Now, endosymbiosis is another idea in which cells were proposed to have engulfed other cells without digesting them. So these other cells, so you could say a larger prokaryote. Typically, if you're a larger prokaryote, it's a little bit more difficult to consume you. And if you happen to engulf a cell that happens to have some useful metabolic processes, such as the creation of ATP from the breakdown of glucose using oxygen in an aerobic process, if you simply don't digest it, well, you might be able to reap some of the benefits of that ATP production. And then that smaller bacteria, well, if you're able to continue feeding it substrate in order to make that ATP, well, now that smaller prokaryotic cell has an advantage in the sense that it is not predisposed to becoming consumed by other prokaryotic cells, prokaryotic species. Well, eventually what happened is the larger eukaryotic, the early eukaryote, lost their autonomy, meaning they became dependent on that energy production from early mitochondria. And that early mitochondria probably lost their autonomy in the sense that if they were, pushed outside of the eukaryotic cell, they could potentially die to their harsh environment. But as a result, the larger eukaryotic cell, with that boost in energy, they were able to devote some of that energy into forming organelles. So here's just kind of a overview of a few of the events that may have happened. So first you get infoldings of the plasma membrane of an ancestral prokaryote, which gave rise to endomembrane compartments. So those are going to be things like your nucleus, your nuclear envelope, as well as your endoplasmic reticulum. And then in the first endosymbiotic event, so that's just going to be consuming something and then having some form of a symbiotic or codependent relationship on one another. The first endosymbiotic event, the ancestral eukaryote, so that larger cell, consumed the consumed a type of aerobic bacteria that eventually evolved into mitochondria. And so those mitochondria, like I mentioned earlier, or early mitochondria, were smaller prokaryotes that happened to be capable of producing ATP using some kind of substrate and maybe the use of oxygen, well, aerobic implying the use of oxygen. So then in the second endosymbiotic event, the early eukaryote consumed the photosynthetic bacteria, a type of photosynthetic bacteria that would have eventually evolved into chloroplasts. Now, one thing that I should mention is oftentimes people get confused by the presence of chloroplasts in certain organisms. They assume that because they contain chloroplasts that they do not have mitochondria. So oftentimes people think that because you're a plant, you do not have mitochondria, but in fact, all plants that have chloroplasts will also have mitochondria. Those mitochondria are there to take the substrate that your chloroplasts create, which are sugars, and when the plant needs it, that mitochondria can break it down for energy. So eukaryotes have a membrane-enclosed nucleus in each of their cells, and eukaryotic cells tend to be about 10 times larger or more, or 10 times larger or even larger than many prokaryotic cells. And their compartmentalization is what allows for those size differences. So let's check out this scale for different particulates as well as cells, showing their relative size to one another. So here we just have some viruses, and eventually we'll make it to our first living organism, which is Staphylococcus, a bacterial cell. Same with Lactobacillus, E. coli. Then we have red blood cells, which are devoid of nuclei, Baker's yeast, which is a type of fungi containing a nucleus, so it's a eukaryote. We have sperm, which use whip-like flagella for their motility. They're also haploid. We've got neurons, human eggs, euglena, which is a type of protist, which is a single-celled eukaryotic cell. Same with diatom, same with paramecium, same with amoeba, which use pseudopodia as well as cytoplasmic streaming for movement.